Thank you very much, Jerry. I will say that so much is related to the environment that I am brought up in, and that we have this tradition in Scandinavia, and I feel myself just being one in, say, a long line of very good researchers, and they are newcomers, and the new generation, they have really contributed to where we are standing today. And without that, I wouldn't have been able to function and act the way I have been able to do over the years. In addition, I will say I feel very privileged. As Jerry pointed out, I have been around since 1960 approximately, and that's a long time. And then having a chance to share my ideas, some data with you, this is also a privilege, and I thank you very much for that. Now, coming to what I had planned to talk about, it will, of course, be very much related to top athletes, especially then they being the subjects. But when it says here, contribution to the understanding, that's probably right. But then I was thinking a little bit, have I contributed at all to help these athletes to become better? And I questioned that. I got a little bit of a relief last night when Scott had his presentation of being a manager coach for top athletes, when he said that it was important to have people in basic science who sometimes could guide or help the people who are more directly in contact with the athletes. And on that, say, level, I have had a chance both to work with the Swedish Olympic Committee since the 60s and with, it's called in Denmark, Team Denmark, that's the equivalent. And that has been extremely helpful because, as Scott said, many of the problems that we have studied, they come from conversations, discussion, and trials on these top athletes. But the goal from my side has been to try to understand human, say, physiology, especially then using exercise as the intervention. Now, the picture you see here, that's from France. You may not know it, but there was a Frenchman doing the first stationary bicycle, and it was basically the same principle as you see here. And that's also then from France, early in 1900, a little bit more sophisticated setting. But I will not continue on the French line. Being in being here in London, and then the Olympics coming up here, I think the right thing is to have, say, Hill as a guidance through this presentation. And I would say there is no top scientist who has done so much to help us to understand what is, say, the basis for human performance, both on the muscle level as well as on the systemic level and the cardiovascular level. And I will return to that several times during the talk now. Here you can see some of his early experiments where he studied acceleration in humans, and he also then, together with Lupton, made this what I call Hill's wheel, where you could then measure dynamic strength. And he produced the first force velocity curves with this setting, and it was very, very accurate. And he also then determined time to peak force. The reason for showing these data and bringing it up to you tonight, that is to have a chance to then bring this quote to you. Because, you know, he got the Nobel Prize 1923, and that was not for this work on humans, it was primarily for his work measuring heat in the muscle contracting, and by that then demonstrate some of the early metabolic events, making it possible for the contractile machinery to get force or tension developed. But you may have had the chance now to read it while I've been talking, but I will read it to you. As it proved, work or hard muscular exercise in man provided the same opportunity of getting accurate and reproducible results as is found in experiments on isolated muscle. Indeed, in some ways, man was the better experimental object. When trained, he can repeat the same performance again and again. And it reminded a matter of always of satisfaction, sometimes even of excitement as the work evolved. To find how the experiment on man and those on isolated muscle confirmed and threw light in one another. I think today we are missing very, very many who are focusing on various species all the way down on the subcellular and molecular levels, and sometimes you wonder if it really is linked to then the understanding of how the human function and perform. They should, I think, be more aware of Hill's view. It would be helpful. Now, before all these fancy machines came to measure then dynamic strength, we used the Hill's wheel some 30 years ago, and it was very, very helpful because we wanted to see just these force velocity curves in humans and how much the individual variation was. And as you can see here, these are 8 subjects, and you have the velocity on the x-axis and the force developed on the y-axis, and 8 subjects, and they are very, very different. And what is the explanation? Yeah, the explanation is that they had different fiber composition in their biceps brachii muscle, and if you then took into consideration the area of type 2 muscle fibers, then you can see how good the relationships is. So that's point then in the direction of the different fiber types having then, say, a speciality. The type 2, of course, then for quick strength development, and the type 1 more on the endurance type. But now we have data, I've used here Bottinelli's, on single fibers of the different types, the pure and those co-expressing the different myosin heavy isoforms. And you can see up to the right how closely related the Vmax is to what fiber type that has been studied. And then you can see also the force velocity curve, and then you have to the right the power, and there is really a difference between these fiber types, as you all are well aware of. But that's not really why I'm bringing up the subject. It is now that I think people are too influenced by this beautiful graph. Burke produced it in the late 1960s, and he used cat as the subject. And there is nothing wrong with all these data, but it was then coined more or less that a type 1 fiber was oxidative, the fast twitch 2A fiber that was both glycolytic and oxidative, varied a little bit which was the highest, and then definitely the type 2B or X fiber, depending on species, was glycolytic. And that is not the case, that there is this very, very close coupling, or almost say fixed coupling between myosin heavy chain isoforms and the metabolic capacity. And I hope I will, with a couple of slides here, convince you that that is not the case. Erik Rick talked yesterday about forgotten articles. I think this is one example. Eva Jansson and Lena Kaiser in Stockholm, they did study runners, very good runners. As you can see, the lowest value in the group was 72 mLs per kilo. I can say the mean value was 77. And they were in top shape, these runners, when the biopsies were taken. And if you compare that with the sedentary, okay, the sedentary, there is this higher oxidative capacity in the type 1 fiber and the lowest in the 2,8X fiber if they were co-expressed in the two isoforms. But have a look on the runners, and that's the gastrocnemius muscle, the same fiber types, and they are 4, 5, 6 times higher, and there is no significant difference between the 2AX compared with the type 1. So the metabolic capacity is not primarily dependent upon which myosin heavy chain isoform. 2002 was this article in Nature that gives a further support for that there was a close coupling. They had transgenic mice overexpressing PGC1 alpha, and you can see both on the pictures above and to the left that they really have these typical oxidative, say, drive. I mean, the PGC1 transcription factor is really causing mitochondrial biogenesis and also other signs as myoglobin. But what they also saw was that there was some formation of 2A fibers to type 1 fibers. And they then concluded that the main reason for the increase in the oxidative capacity was the increase in type 1 fibers, and PGC1 alpha was then inducing those gene changes. Frank Booth, he was not happy with the conclusion, and he checked the original data very carefully and wrote an article about it, and the response then to Lin et al.'s conclusion was that he pointed out that the elevation in type 1 fibers was less than 5% as compared to 200 to 300% elevation in mitochondrial capacity. Very few have tried to follow up on it, but Hanshin, he is active in Basel, but he has spent a couple of years together with Spiegelman in Boston. He has an article late 2011 in American Journal of Physiology where he checked on the PGC1 alpha and if there was a chance that there was something in the cytosol that could, say, modulate this dual effect, both on mitochondrial biogenesis and affecting the genes encoding for myosin heavy chain. And he found that there was a link to the calcium, the cytosolic calcium concentration, and depending upon the type of stimulation, there was then different concentrations, and that then caused a blocking, or at least, or even, we'll say there was an impairment, sometimes a full blocking of then activation of the myosin heavy chain 1 gene, or the one encoding for that protein. And I think Stefano, he alluded to this yesterday in one question he got, that the calcium here has maybe that effect, and explain then why there is not this close coupling. And it also then confirm in a way what also Stefano talked about yesterday, Eccles and Buller's early studies with cross innervation, and there was the fire pattern, and then the intracellular calcium concentration. That was a key to whether or not these myosin heavy chain genes were activated or not. Now, to then take this a little bit further, you can have a look on this. It's not easy to see which species or which muscle it is, but what you will see here is a lot of then mitochondria. And the answer to the questions there, they are that it is muscle from man, and it is triceps brachii, and it's a type 2 fiber. So this is further, if you say so, confirmation that a type 2 fiber can be very, very oxidative. It's just a question of how it is used. And the biopsies here, they are taken from Norwegian cross-country skiers. They are better than the Swedes, and they are also friendly enough to let us take biopsies from them. And I would say they are good. 3 of them, they were just becoming seniors when we did this study, but 3 of them have taken gold medals in the latest Olympics. So it is the top guys. But you can see, you know, the triceps brachii, the extensor here, used a lot, of course, in skiing. They have the highest ever recorded mitochondrial volume. It's the different subfractions is the same as you see here for the largest fraction, the myofibril. Now, if we take it a little bit further here, the RACING CAMEL. You may not be aware of it, but they run easily 40, 42, 44 kilometers an hour, and they can do it for half an hour or longer. So they are quite fast as a horse, and they have more of an endurance than most horses have. Horses then being able to have that high speed. Now, we have taken lots of biopsies from them, from these camels, and one thing then, if we take a human vastus lateralis with about 50-50 in fiber types, then the biceps femoris is the same in the camel, and you can see that the electrophoretic pattern is very, very similar. But if you now look on triceps from the camel and take the vastus lateralis from the camel, the triceps has also very many type 2A or X fibers. They are highly highly oxidative. Citrate synthase, the beta and HED enzyme for the beta oxidation of fatty acids, and higher than you can see then in the rather rich in type 1 fiber, the vastus lateralis on the camel. But the next step there is what the fiber, this type 2AX, is high lactate dehydrogenase, high phosphorylase activity. So it's high both glycolytic and oxidative, and it's a type 2AX fiber. So this is just example that I hope you will remember, and then closing this part, then you should then take home the following message. The bottom part here, ATPase staining of fiber types, a sprinter, a pretty good one, and you can see pretty good marathon runner. They have very different fiber type compositions, but that's then probably inherited. But what you see on the top part here, this is oxidative staining. If we have had also a glycolytic staining, you would have seen the reverse then for the sprinter and the marathon. So the upper part then, that is really what has been developed by proper training. Now, another quote from Hill and Hill-Lupton. It's probably the most, say, commonly cited and discussed, say, quote from Hill and Lupton. However much the speed be increased beyond this limit, no further increase in oxygen intake can occur. The heart, lungs, circulation, and the diffusion of oxygen to the active muscle fibers have attained their maximal activity. Hill may not have had the best, say, leveling off data when it comes to maximal oxygen uptake, but he definitely did see what the limitation was. In Scandinavia, there were quite a few quite active top researchers who were also then interested in classical exercise physiology. Here is just to demonstrate how they, in the late 1910s, measured both oxygen uptake and cardiac output, tethered swimming, tethered rowing. But the same group, they had a little bit earlier also done similar studies when running. And you can see it's the higher the speed to the right, that it starts to, say, level off. And if you plot data in the table for the two, studies from that time, usually n equals 2, and the same persons being subjects, they are also the authors of the paper. But have a look. I mean, that's data from 1918. And if they had plotted their data along that line, they would have been those who would have defined the maximal oxygen uptake, the first then in the world. But they didn't realize what they had found out. But it was Hill who then saw the data and then had seen what the peak for maximal oxygen uptake was, and then increasing the speed, it didn't help to increase maximal oxygen uptake. Now, then the question, of course, that was discussed and also then used as the basis for a study by Niels Secker, a classical study in 1977, was that can the heart supply all muscles with an ample blood flow in whole body exercise? And when Niels Secker defended his thesis, where this was part of it, there was, you wouldn't imagine, it was a discussion that it couldn't be right. Because in those days, and that's not that long back, one thought that the skeletal muscle peak flow was around 50 mL per 100 gram a minute. And then when the muscle started to contract, there was vasodilation. Local factors caused that, and then the heart just pumped a little bit more and filled him up. And I mean, it was no question that he got, his thesis was accepted but it was really thrilling to hear the discussion, and it really inspired me and the people around me, and we have spent lots of time then on it. Nils Sechter is a rower, so we thought of using rowing because there you have also lots of upper body work, but you bend your legs, hip too much for being able really to do good measurements with catheters in the vessels of the leg. So we turned to skiers to then have a natural form of physical exercise where upper body and legs were both involved and heavily involved. And the good thing is that you have roller skis, and they can then ski on an ordinary treadmill. But of course, being subject in our lab, there are a lot of catheters to be placed before they can start to exercise. And in this case, we had one up in the subclavian vein to measure flow and AV difference over the arm-shoulder region. We had one in the right side of the heart for direct FICT and for cardiac output, and then catheters in both the artery and the vein in the area of just under the inguinal ligament, so we could get flow and AV differences there as well. And I'm showing now this because just seeing that we could then have a pretty good check on all different parts of the body, especially the limbs. But as we had all those data, then we could also estimate the flow and oxygen uptake of the torso by subtracting arm, shoulder, legs, then values from the systemic value. Now, this is the way it looks. Many have probably seen these pictures, but those who know a little bit about what cross-country skiing looks about, this is the classical style, and you can't say differ the way they handle it on the treadmill from when they are on ski and snow. That's the diagonal, and then the other one here, that's the double poling that's taking over more and more, especially in skating, and it's a very efficient way to get high velocity. Anyway, the first question then, what happens if you compare peak arm flows and peak leg flows with what they could attain when they were doing the classical style, both arms and legs as much as possible. And then you can see it dropped both to the arms and to the legs. I thought that there may have been a priority based on Jerry's data with the respiratory muscles, where he nicely demonstrated with it increased work for the respiratory muscles. They did steal then flow and oxygen from the exercising legs. So I thought it was a hierarchy. Heart always got whatever they could get, the respiratory muscles, and then the legs, and then the arms. But there is similar reduction to both limbs, exactly the same, say, relative drop. So there is no, at least in our hands, no difference then between the limbs. But with all the cats, we could also check on arterial oxygenation, and then coming back to all the great data that they have then collected, especially around Jerry, and we heard about it yesterday from Amman over 2 days ago. You know, either Scandinavians or definitely skiers, they do not desaturate. And if we go back and look, Björn Ekblom has done a lot on top athletes early days with Lars Hermansson, and when they are running, they still keep up saturation. And the skiers, they definitely do. You can see the diagonal, they have a little bit more of a drop, but it's not very dramatic. And with double pooling, you know, up here with their arms, they have also, at maximal exercise, very, very little drop in saturation, and definitely not in PO2. A bit easier way to look at it is here, where you have then on the horizontal the oxygen uptake in liters per minute, and then on the other axis, the vertical one, you have drop in saturation from rest. And you can see at 90%, there is a drop, but it's not more than 2%. And if we go up to the maximal, the diagonal, there is 3, some 4%, but with the double pooling, it's just a straight line. So it's not much of a desaturation. Down in the corner to the right, we can then at least get an estimate for what the oxygen cost is for respiration, because we have this then possibility to get the value for the torso, and then we can subtract heart and other inner organs, and the rest then to respiratory muscle. And you can see how low the values is, especially with the double pooling. And I think, you know, so much of then the raising of the upper part of the chest, giving good, say, ventilation of the lungs, as well as then a good match between ventilation and perfusion, will be part of the explanation. And of course then all the work with the arms, and then also then some of the upper chest muscles, that will also create speed, propulsion forward. And I talked with Jerry about these calculations, and also the low values here, and he didn't scream, but and I have heard rumors that he have said in lectures that they have some odd values in Copenhagen, but yeah, it's anyway. There are, you know, sports like then skiing, where you have extremely high oxygen uptakes, ventilations 200, 250 liters per minute and so, and they do not desaturate. But it's very special the way the arms contribute, I think, then also to the respiratory activities. Now the last part then of this is then oxygen extraction. And I have a little bit different view from quite a few in regard to the role of mitochondria. If you have a look to the left there, they have been inactivated for a couple of weeks. And then you can see the venous return of oxygen. That is the lowest that we have here when you compare with sedentary and those who have trained a couple of months, years, or are elite athletes in endurance sports. So the lowest there, venous return, is in the inactivated, having probably less with mitochondria, and the reason then that they extract that well, that is that if you look on the top there, the number of capillaries is low, but their muscle blood flow is very, very low. And as you will see in the following slide, the mean transit time is then not very short, in fact quite long, and the mean transit time, I think it is what counts. So if we look then on the mean transit time, they are estimated, but if anything, I think they are on the low side. If you don't believe in those types of, say, estimations, you can just see, mean transit time, that's nothing else than dividing capillary blood flow with muscle blood flow. And if you check on the capillaries, they increase more with training than what the muscle blood flow does. So mean transit time must go up. And the key reason then for then having this very good extraction is not that you have more mitochondria, it is that you have a longer time for the erythrocytes to deliver the oxygen and that taken up then by the muscle fiber. Now, you have from my way of presenting these things, you could ask if I am also making, say, the same thing like Hill is pointing at here. The background for this quote from his last book, it's odd how one's brain fails to work properly when pet theories are involved. I mean, in his case, it was that he did stick to some explanations for his findings on heat production in the muscle that later was not only debated but also probably not right. And he didn't give up to fight for his views. And sometimes I have been looking at this and wonder if I am just as stubborn as Hill was once upon a time, and just trying to defend that the heart is limiting and its mitochondria, their role, of course, is to use the oxygen, but they are not the limiting factor. They're the reason for an enlargement of the mitochondrial content and the oxidative capacity, that's primarily for metabolic reasons, substrate choice, and improve that from carbohydrate to fat combustion. Now, then changing the last part of my presentation, I got inspired by Ben Levine when he talked about the heart and the large or the importance of diastolic function and then end diastolic volume. And as Jared mentioned, we did this Dallas bed rest and training study, and if you look carefully on the heart volume, stroke volume relationships data, you can see that they are very close to when they started the study to that line, and then when they were trained, they are directly on the line. But after the 3 weeks of inactivity, they are definitely not producing the stroke volume that they should be able to do with the heart volume that they had. So the question that we raised already in the 70s, but we haven't done enough about it, so it's not solved. But I thought it's worthwhile then to say a little bit about the question that is raised. Does the training status of the exercising muscle play a role for the size of the stroke volume? And we did then a study published in 1991, one-legged training, as you heard a lot about from Erik Richter, and then they performed here also one-legged exercise. And you can see that when they are exercising with the trained leg, they have a larger end diastolic volume than when they are exercising with the untrained leg. And this is a case, the same body, the same heart, and it's maybe 15 minutes between if they have exercise with the trained or with the untrained leg. So the same absolute workload here elicited quite different heart rate, lower with the trained leg and that is compensated for by a larger than end-diastolic volume, which creates then the larger stroke volume. And it looks, if we just take the data that we had from that study, you can see ejection fraction is about the same, but the difference is the end-diastolic volume, and you can see the difference in the heart rate. So that paper was published not in a very good journal, because the reviewers said, yeah, but you have to then compare with the same heart rate. So because here, you know, you have a lower heart rate and then a longer diastole, and you don't have central venous pressures on the, sorry, you have central venous pressure that is on the right side, but you don't have the pressures, filling pressures for the left side of the heart. So it took some time, but we got then also the ethical permission to do pacing studies. So we have our subjects, they train one leg and the other one to really make the difference. They were that was really inactivated the untrained leg with a cast for some time. And then the study checking the trained leg and the untrained leg, and before they had also then both the catheter in the pulmonary artery as well as then a pacemaker introduced to the atrium on the right side. So I'm just giving you one slide of results, and you can see there, if you go to the right, that's the best one. There you have the untrained, and then you have the trained, and then you are pacing, and you can see that the wedge pressure is a little bit higher without the pacing compared then with the untrained leg, but it's not significant, that small difference. But the key finding here, you have also on the left side, when you have a time for the diastole that's equal to the untrained leg, you have the larger end-diastolic volume, and with the consequences then also how it affects the stroke volume. So that's where we are, and it's really so that in one way or the other, the heart feels or senses, if we use a more, say, scientific term, that it's a trained muscle. And how that occurs, we unfortunately still don't know, but we think that there are receptors then on the cardiac myocytes, and either nucleotide or epinephrine will bind to that, and that's then different than plasma concentrations. And we know that the titin and the elastic component of the sarcomere is sensitive to NO. So that's one hypothesis, and the good thing is that it can be studied because both the beta receptors as well as the P2Y receptor for ATP or other nucleotide, they can be blocked, at least some of the P2Y receptors. So we are looking forward to the results on that. If I'm part of the study, or I don't know, but hopefully someone will do it. But I will also then point to what Claude Bouchard touched upon in his presentation. He talked about candidate genes for then aerobic fitness, and he on his list, titin was then highly ranked, and that's a very complex gene. And also the Claude Bouchard group, they have data demonstrating that after training, there is then more common with the larger improvement in stroke volume and drop in heart rate if they have a special mutation in that very, say, difficult gene to study. So I think that is an eye-opener to me, and I hope also for you to be interested in this specific problem that Titan is to go for, and also not only then the gene, say, variation that may exist, but just as much on how that is regulated in the heart. And that, I think, will give us an insight to what really makes the stroke volume to be so large with training and the best top athletes. But those studies are to be done. Now, coming then to an end, because Jerry brought it up, and I feel that I'm just a product of this Scandinavian tradition, and then special circumstances, because I was just a teenager when August Krogh and Johannes Lienert died. But I was fortunate to have one of their best students as my mentor. He moved to Sweden to establish this field in the 1940s, and when I started in the field, 1959, part-time, and soon after full-time, I saw him every day, and he gave all the knowledge he had and all the inspiration. And he also then was one not pushing me, but asking me if I shouldn't move to Copenhagen, where the Mecca is for this field. And then I succeeded Marius Nilsson in Marius Nilsson in the lab, and then had the chance to work along with Erling Asmussen for several years. So that's my base, and without that, I don't think I had been able to do what we have in a large group been able to accomplish. Now, I would like then to finish with a thing that Erik Hove Christensen said just when he retired, and he was chairman for the last session at a biochemistry of exercise meeting in Maglingen 1974. And what he said: "I envy you. My generation of researchers have described several of the acute and chronic responses to exercise. You have the tools, the methods, and the knowledge to unravel the mechanisms by which these processes are regulated. Remember, not until we understand the regulatory mechanism, our research goal has been reached." And I'm sad then to say that 40 years later, I feel that what I, to a large extent, have been doing, describe more, say, phenomenon together with exercise and the human being. But we have maybe identified some possible mechanisms, but there is really a long way to go, and that's for especially the young people in the audience to really get going. And I hope when you are ready for retirement 30, 40 years from now, you don't have to stand up like I to say, sorry, I have also spent most of my time describing. Thank you very much for your attention.